Welcome everybody to AP Daily. This is AP Biology topic 6.1. We are going to be talking about DNA and RNA structure, how heritable information provides for the continuity of life. And the skill we're working on today is explaining biological concepts. So what are our goals? What are we going to learn? We are going to try to attempt to answer the question, what is the source of heritable information found in cells? What are the characteristics of DNA and RNA that allow them both to be used as hereditary material? And what differences exist between the heritable information found in prokaryotes and eukaryotes? So let's dive into it. DNA, and in some cases RNA, is the primary source of hereditary material. So what we're talking about here is that genetic information is stored in a sequence of bases. Those are bases in either DNA or RNA, and that allows us to transmit that genetic information from one generation to the next through that DNA, or in some cases RNA. This DNA is packaged into chromosomes. That's what the picture is showing you over there. And those chromosomes are then passed from parents to daughter cells. We're going to talk a little bit later, but viruses specifically use RNA molecules to encode their genetic information. So again, DNA and sometimes RNA is going to exhibit specific nucleotide base pairing. So I mentioned before that we have a specific sequence of bases. Those bases pair with each other. So DNA and RNA are going to be structurally similar in that they're both polymers. They both contain nucleotides. Those are arranged in chain-like molecules, and they both follow base pairing rules. When DNA nucleotides pair with each other, adenine is going to pair with thymine. That's what you can see down here, yellow and green. And guanine is going to pair with cytosine. So that's what you see, the blue and orange. When RNA becomes involved, when RNA nucleotides pair with these DNA nucleotides, we're going to see one change. And that change is uracil. So the RNA's uracil nucleotides are going to pair with DNA's adenine nucleotides. This base pairing is conserved through evolution. And what I mean by that is these base pairing rules are they're specific. They happen between these purines and pyrimidines, and all organisms use the same rules. Our pyrimidines, those are the ones circled over here in purple, they have a single ring structure. So we've got uracil, cytosine, and thymine. You'll notice in the middle of each one, there's one ring with some things hanging off of it. And then we have our purines, adenine and guanine, and those are a two-ring structure, a double-ringed structure. So prokaryotes and eukaryotes are a little bit different. Their genomes have a couple of different aspects that we want to talk about. Prokaryotes typically have circular chromosomes, whereas eukaryotic organisms have multiple linear chromosomes. The prokaryotic genome is typically smaller than a eukaryotic genome, and prokaryotes and eukaryotes can contain plasmids. Those are small, what we call extrachromosomal, double-stranded circular DNA molecules. I know that's a mouthful, but think about it as just a little bit of extra DNA, a little tiny circular extra bit. In prokaryotes, those plasmids are found in the cytosol, while in eukaryotes, those are found in the nucleus. So let's practice this. Let's put this into a problem and see what we understand. The skill that we're focusing on, remember, is concept explanation. So we're going to be explaining biological concepts, processes, and/or models in an applied context. So I have a question here for us to look at. What I'd like you to do is to read over this question, pause the video, take some time to digest the information, pick what you think is the best answer choice, and then hit the play button again. Come back to me and we'll see how you did. All right, welcome back. Hopefully you had a chance to digest what is going on in this question. We are given a figure. That figure contains relative amounts of nucleotides, A, T, C, and G, so adenine, thymine, cytosine, and guanine. The question tells us that genetic material found in two different organisms is analyzed to determine the relative amount of uracil nucleotides contained in an RNA molecule used to copy DNA information. The RNA molecule found in one organism contains 15% uracil, and the RNA molecule found in another organism contains 30% uracil. Which of the following statements best explains, that's our skill, why the RNA molecules belong to different organisms other than those represented in figure 1? So do a drum roll in your head, and hopefully you came up with answer choice B. The relative amounts of adenine in DNA should be the same as the relative amounts of uracil in the RNA used to copy. Remember, adenine and uracil are going to have that base pairing. So what are our takeaways? What are the big things I hope you learned from spending these couple minutes with me? First, DNA, and in some cases RNA, is the source, the primary source of heritable information. Second of all, DNA and RNA store information as nitrogen base sequences. Base pairing occurs when specific pyrimidines pair with specific purines. And this base pairing is conserved through evolution. All organisms pair in the same way. And last but not least, our prokaryotes. They typically have circular chromosomes, while those eukaryotes have linear chromosomes. The prokaryotic genome is typically smaller, and those prokaryotic plasmids are found in the cytosol, while eukaryotic plasmids could be found in the nucleus. I hope you enjoyed spending a little bit of time with me today. If you have any questions about it, feel free to go back, rewatch the video. Thank you for your time. Hi, everybody. Welcome to AP Daily. This is AP Biology topic 6.2. We are going to be talking about replication, focusing on mechanisms used to copy and transmit genetic information. The skill we're going to be using today is explaining relationships between biological concepts. My name is Lisa Niesman. I am coming to you from Brooklyn, New York, and let's dive in. So what are we going to learn today? What questions are we going to try to answer? First of all, what is the purpose of DNA replication? What does it mean for this replication process to be semi-conservative? How does the directionality of a DNA molecule influence the replication process? And lastly, what are the specific enzymes involved in replication, and what is the function of each of those enzymes? So let's dig into it. The purpose of DNA replication is to ensure the continuity of hereditary information. And what I mean by that is we have to copy this genetic information during our DNA replication stage. This occurs before cell division. And by copying it, that allows transmission of a complete genome from one generation to the next. So we have to make a copy so we can pass on a copy to the next generation. There are quite a few specific mechanisms that ensure the continuity, that make sure each copy that we have is going to be the same. So there's a word that we used when we're talking about DNA replication. We say that it is semi-conservative. And what this means is that when we have a double-stranded molecule of DNA, when we make a new copy, each of those original strands is going to serve as a template for the new strand. So our new or complementary strand is going to be pairing with one of those original strands. In this picture or figure that you see, we started off with blue DNA. We've colored it blue in this picture. And if you look all the way to the end, we have 2 copies, but in those copies, one strand is blue, one strand is orange, showing that in the new DNA, one is that template and one is a newly synthesized or complementary strand. So I mentioned directionality. The directionality of this molecule really strongly influences the replication process. We know from previous lessons that the DNA strand has a terminal phosphate group on one end and a terminal hydroxyl group on the other end. We have names for these. So we refer to that phosphate terminus as the 5' end, and we refer to the hydroxyl terminus as the 3' end. So the phosphate terminus is 5', the hydroxyl terminus is 3', and we usually denote those the way this picture has it as sort of 5' and 3'. You'll see that notation. These two strands, the 2 strands of DNA, run anti-parallel to each other, meaning they're running in opposite directions. So the 5' end of one strand is opposite the 3' end of its complementary strand. As we're adding nucleotides, so we're replicating, we are making a new strand of DNA, they can only be added in the 5' to 3' direction. So what this means is that one strand is always going to be made continuously in one long strand. We call that one the leading strand. The other one then is going to be synthesized discontinuously, meaning in little fragments, and that's called the lagging strand. So as we're synthesizing new strands of DNA, one of them is always going to be called the leading strand, and the other one then is the lagging strand. There are quite a few enzymes involved in this process. There are 4 of them that we're going to focus on. The first is helicase, and helicase's job is to unwind those DNA strands. It's going to take the helix of DNA and unwind it. The second one is topoisomerase. So topoisomerase's job is to relax what we call the supercoil at the replication fork. It's a fun sentence to say. The replication fork is the V. It's the sort of crux where the 2 strands are separated. And topoisomerase's job is to hang out there and relax the supercoil of the DNA. DNA polymerase, you'll see it has quite a few jobs. It synthesizes the new strand of DNA. So it is sort of the workhorse enzyme. It's going to do the bulk of what we're talking about. It requires RNA primers to initiate that synthesis. It attaches to the 3' end of the template, and then it builds its strand, like we mentioned before, in the 5' to 3' direction. Last but not least, we've got ligase, and ligase's job is to join those itty-bitty fragments on the lagging strand. So remember I said the leading strand gets synthesized in one long, beautiful strand. The lagging strand has little bits that need to be stuck together. And so it's ligase's job to stick those strands together. So let's put all of that to practice. Let's look at a question and try to synthesize what we've learned. Again, our skill focus for this is visual representations. So we're going to be explaining relationships between different characteristics of biological concepts, processes, or models represented visually in applied contexts. So you're probably going to get a picture. So here's that question, the promised picture. Pause the video, explore this question, come up with what you think is the best answer, and then come back to me and we'll see how you did. All right, welcome back. Hopefully you had a chance to explore this diagram, see some familiar terms and notations that we talked about, and hopefully you had a chance to mark the correct answer. So we've got a figure, it's illustrating a model of molecules involved in DNA replication and their placement relative to each other. Our question is, which of the following statements best explains how topoisomerase and helicase are functionally related to each other during the process of DNA replication. So drum roll and D. Helicase unwinds the original strands as topoisomerase relieves coiling tension ahead of the replication fork. So we're looking at the functions of those two enzymes and how they work together. So our big takeaways, what did we learn in this video? We learned that DNA replication ensures the continuity of hereditary information, which allows us to transmit genetic information between generations. We learned that DNA replication is semi-conservative, meaning one strand acts as a template for the new or complementary strand of DNA. We learned that DNA is synthesized in the 5' to 3' direction, which leads to a continuously synthesized leading strand and a discontinuously synthesized lagging strand. And then we had our 4 enzymes: helicase, whose job is to unwind the DNA; topoisomerase relaxes that supercoil; DNA polymerase synthesizes the new DNA strands; and ligase works on that lagging strand to join the DNA fragments together. Hope this video was informative to you. I hope it clarified some topics. If there were questions, go back and rewatch the video. Otherwise, thanks and have a nice day. Hi, everybody. Welcome to AP Daily. This is AP Biology, topic 6.3, transcription and RNA processing, daily video number 1. This lesson is going to focus on transcription and RNA processing. The skill we're going to be working with is explaining relationships that are represented visually in applied contexts. My name is Lisa Niesman. I am coming to you from Bay Ridge Preparatory School in Brooklyn, New York, and let's get started. So what are we going to learn today? We are going to tackle a couple questions. First, what is meant by the flow of genetic information? What is transcription? What are the specific mechanisms involved in the transcription process? And what are the different types of RNA molecules, and what is the function of each? So let's get started. Genetic information flows from DNA to RNA to protein. Our genetic information is typically stored in DNA molecules. RNA molecules are then used to facilitate protein synthesis using the DNA information. So you can see in the picture, they kind of come at that middle step between the DNA and the protein. And ribosomes contain RNA, and they assemble the proteins. So transcription is the process in which an enzyme directs the formation of an mRNA molecule. So there's a lot of information, a lot going on here, but our key ideas is that the DNA strands are going to become separated during transcription. One strand is going to serve as a template, which is also known as the non-coding strand, minus strand, or antisense strand. All 3 of those terms mean the same thing. That is our template strand. The other, or non-template strand, is also known as the coding strand. So the strand that's going to serve as the template depends on the gene that is being transcribed. The gene that is targeted for transcription is therefore located on what we call that coding strand. What happens then is the enzyme RNA polymerase is going to synthesize messenger RNA. We usually refer to that as mRNA, lowercase m RNA, in the 5' to 3' direction by reading the template in the 3' to 5' direction. So the RNA molecule is a transcribed copy of a particular gene, of the gene that we want. So by separating these DNA strands, identifying the gene that we want, we're then able to make an mRNA copy. We're able to transcribe that copy of the gene that we want. It's about RNA, and there are three types that I want to make sure to distinguish and explain for everyone. So messenger RNA, or mRNA, that's the one we were just talking about. That carries the genetic information that was contained in the DNA. It carries it to the ribosomes. That information is then used to direct protein synthesis in that ribosome at the ribosomal site. And within the RNA, we have a notation called a codon, which is a 3-base sequence found on that mRNA. So each group of 3 nucleotides we call a codon. The second one is transfer RNA, or tRNA, and that is recruited by ribosomes, which are going to help create a specific polypeptide as directed by the mRNA. So each tRNA carries a specific amino acid, and there's a sequence on the tRNA called the anticodon, which is a 3-base sequence in that tRNA. So the correct base pairing of the tRNA anticodon with the mRNA codon is going to result in the release or addition of an amino acid to the growing polypeptide. So we need that codon-anticodon pairing. The last type of RNA molecule we're going to focus on is ribosomal RNA, or rRNA. These molecules are what we call the functional unit of the ribosome. They're responsible for protein assembly, and this is where that pairing of codon to anticodon occurs. It occurs in the ribosomes, which then allows us to create these primary polypeptides as the tRNA brings and then releases its amino acids. So that was a mouthful, but let's put it into practice. Let's see how you guys are doing with this. So the skill that we're working on again is visual representations, which means we're going to be asked to explain the relationships between different characteristics of biological concepts, processes, or models represented visually in applied contexts. So there's our picture. Look at that figure, read the question, try to come up with what you think is the best answer, pause the video while you do that, and come back to me when you think you have your answer. Welcome back. Hope you enjoyed your time looking at figure 1, which is a model of the process involved in gene expression. So our question tells us that we're looking at transcription. It tells us that transcription is a process involved in gene expression and includes the use of enzymes. This figure represents the transcription process. Which of the following statements best explains how RNA polymerase is involved in the transcription process? So hopefully you guys have C written down on wherever it is that you're taking notes. RNA polymerase's job is to copy the gene by base pairing with that non-coding strand. So hopefully you got that question right. Hopefully you're feeling confident and comfortable with the content that we talked about. If not, feel free to go back, rewatch the video, take some notes for yourself, and look at these key takeaways. Make sure that everything is synthesizing itself the way we hope it does. So our big takeaways, what did we learn? Genetic information flows from DNA to RNA to protein. This is sometimes called the central dogma of biology. Second, transcription is the process by which RNA polymerase uses the non-coding strand of DNA as a template to produce that mRNA molecule. So it's that first step, DNA to RNA. RNA polymerase synthesizes mRNA in the 5' to 3' direction, while reading the DNA in the 3' to 5' direction. And we talked about 3 types of RNA molecules. Each has their own structure, each has their own function. So the mRNA carries the copy of the gene to the ribosomes. tRNA is recruited to the ribosome in order to build the primary amino acid sequence or polypeptide as dictated by that mRNA sequence. And lastly, we have rRNA, or the functional building blocks of ribosomes, which are responsible for that ultimate protein assembly. So hope that helped clarify some topics for you. I appreciate you taking the time to watch this video, spend some time on AP Daily. Have a great day. Hi, everybody. Welcome to AP Daily. This is AP Biology, topic 6.3, transcription and RNA processing, video 2. You guys came back for more. This lesson, we're going to be talking about the modifications that are made to the mRNA after it's transcribed from DNA. The skill that we're going to be working on is explaining relationships that are represented visually. My name is Lisa Niesman. I am coming to you from Bay Ridge Preparatory School in Brooklyn, New York. Let's get started. So what are our key questions today? What are we going to learn? We are going to start by looking at the mRNA transcript modifications that occur in eukaryotic cells. We're going to we are going to answer the question, what is alternative splicing? And we're going to answer the question, how does alternative splicing result in different proteins? So let's dive into it. There are a series of enzyme-regulated modifications that occur to that mRNA transcript in eukaryotic cells. So one of those modifications is the addition of a poly A tail. It's represented in my picture with the green A's on that 3' end. That's 100 to 200 adenine nucleotides. So a whole bunch of A's stuck on the end. This increases stability, and it also helps with exporting from the nucleus. On the other side of our mRNA transcript, we add what's called a GTP cap. So that's a modified guanine nucleotide, and its job is to help protect that transcript and help the ribosome attach to this mRNA when it gets there. So we have a couple more steps that are going to happen. You'll notice these have that poly A tail, that GTP cap, and now we're going to look at introns and exons. So our introns are the sequences of mRNA that do not code for amino acids. They are excised or removed during RNA processing, and so they are then not included in the mRNA transcript. Alternatively, we have the sequences called exons. So exons are the sequences of mRNA that do code for amino acids, which means they're retained during RNA processing, and those different exons are then connected to each other to make what we call the mature mRNA transcript. So you can see in the picture, we have the primary transcript, which contains both those introns and the exons, and then the mature mRNA only has those exons. I mentioned this term alternative splicing. So the process that I just mentioned of splicing those introns, connecting the retained, those kept exons, is called alternative splicing. And so what ends up happening is that there are a lot of different exons in the primary transcript. In my illustration here, there are 4 different ones. I numbered them very creatively, 1, 2, 3, and 4. But what we can do is we can actually produce different mRNA transcripts from the same primary transcript. And the way we do that is by retaining those exons in different variations. So you'll notice I have transcript 1, and that has exons 1, 2, and 3. Transcript 2 has exons 1, 2, and 4. So those are different exon combinations, which are then going to lead to different proteins. So in my illustration, transcript 1 encodes protein A, and transcript 2 encodes protein B. But remember, those were created from the same primary transcript. So let's practice this. Remember, our skill that we're working on today is visual representations. We're going to be asked to explain relationships between different characteristics of biological concepts, processes, or models that are represented visually in applied contexts. So here is that visual representation. I would like you to pause the video, take a look at this figure and question, pick the answer that you think is the best one. Pause the video while you do that, and then come back to me when you have your answer. Welcome back. I hope you had a fun time reading about these introns and exons. The question asked, which of the following statements best explains why this process occurs? The process that they're referring to is what's represented in figure 1. All that they've told us is that it's part of a process that occurs in eukaryotic cells. So I hope that you guys chose answer A. What is happening in this process? The best explanation. Introns must be removed from the mature transcript because they do not code for amino acids, and only sequences coding for amino acids are then sent to ribosomes. So we have those 2 blue circled introns that are removed, and we're doing that so that they are not included in that final transcript, that mature transcript that's sent to the ribosome. So key takeaways, what do I hope you learned from your time spent with me today? I hope that you have learned that the addition of a poly A tail, a GTP cap, excision of introns, splicing, and then retention of exons are all examples of enzyme-regulated modifications that happen to mRNA. That alternative splicing is when introns are excised from a primary mRNA transcript and the exons are retained. Those are the ones that are kept and then joined together. We can also have different combinations of these exons that can be retained for mature mRNA transcripts. Different exon combinations then encode for different proteins. I thank you for your time today. I hope you found this enjoyable. Any questions, go back and rewatch the video. Have a great day. Hi, everybody. Welcome to AP Daily. This is AP Biology, topic 6.4, translation. My name is Lisa Niesman. I am coming to you from Bay Ridge Preparatory School in Brooklyn, New York. In this video, we're going to talk about how genotypes determine phenotypes of an organism. And the skill that we're going to be working with is representing relationships within biological models. So let's dive in. What are we going to learn? We're going to answer a couple of important questions. First off, what is translation? What are the main steps of the translation process? When does translation occur in prokaryotes? How does an alternate flow of information affect translation in retroviruses? And how does the process of translation serve as evidence of common ancestry? So let's dive into it. Translation of mRNA generates polypeptides. So our main idea is that translation is a process. It's a process by which that mRNA sequence is going to be used to generate a corresponding appropriate polypeptide. This occurs on ribosomes. The differences prokaryotic and eukaryotic, prokaryotes only have cytosolic ribosomes, whereas eukaryotic cells, eukaryotes have ribosomes both in their cytosol and ribosomes that are bound to the endoplasmic reticulum. So there are many steps here. We're going to talk initially about them in this video, and then we'll go into more detail about them in the next video. But we have 3 main steps of the translation process. In prokaryotic organisms, a little bit different. Translation is going to occur while mRNA is being transcribed. So our 3 main steps, we've got initiation, elongation, and termination. So initiation is where we start the process. Elongation is where that polypeptide is going to grow. It's going to get longer. And then termination, where we stop the process. Retroviruses, they're a special case. They do this in a little bit of a different way. What they do is they introduce viral RNA, not DNA, into the host cells. So we've been talking about a sequence where DNA goes to RNA, goes to a protein. Retroviruses start with that RNA piece. They then need a special enzyme called reverse transcriptase, and that's an enzyme that copies viral RNA into viral DNA. So it goes backwards from our normal process of transcription. It's going RNA to DNA. This is a viral enzyme that becomes introduced into the host cell. And once that enzyme converts viral RNA into viral DNA, that DNA can then be incorporated or integrated into the host genome. Once it's integrated, viral DNA is going to be transcribed and translated just like the host's own DNA, except that transcription and then translation of this viral DNA is going to result in the assembly of new viral progeny. We have this genetic code, and the reason that reverse transcriptase, those retroviruses, work is that all organisms use the same genetic code. These translation mechanisms are similar in nearly all organisms. And so the nucleotides that we use to construct the DNA and RNA are common among organisms. We use this as evidence of common ancestry. So we're seeing the same mechanisms in nearly all organisms. Because of this, that viral DNA, RNA molecules, they're chemically compatible with those host cell genomes, and that allows the host cell's own mechanisms, its own translation mechanisms, to work with that viral genome, even though it's not native to that host cell. So let's see what this looks like. Remember, the skill that we're working with today is visual representations, which means we're going to represent relationships within biological models, including diagrams. So here's our diagram labeled Figure 1. I'd like you to take a moment, read, look, interpret this figure, read our question, and come up with what you think is the best answer. Pause the video while you do it and come back to me when you think you have your answer. All right, welcome back. Hope you have chosen the correct answer. So we have a figure. Figure 1 is a model representing the process of translation. And the question is asking us, which of the following statements best represents the relationship between the different RNA molecules involved in translation? So hopefully you chose answer A. tRNA reads mRNA, and RNA adds amino acids to a growing polypeptide. So we are making that protein using our different RNA molecules. This figure should have helped you come to that answer, showing both tRNA and mRNA interacting with the ribosome to produce a growing polypeptide. So our key takeaways, what do I hope you learned from this video? I hope that you learned that translation is the process of generating polypeptides using the information that is carried on an mRNA molecule. I hope that you remembered those 3 main steps of translation, which are initiation, elongation, and termination. Talk more about those in our next video. And then those sort of different cases. So in prokaryotes, translation occurs while the mRNA is being transcribed. And then in retroviruses, for that genome to be translated, viral RNA has to be converted sort of backwards into viral DNA. That viral DNA then gets integrated into the host genome. The enzyme that does this is appropriately named reverse transcriptase, and its job is to convert that viral RNA into viral DNA. The reason this works is that nearly all organisms use the same genetic code, and we take that as evidence of common ancestry for all living organisms. So I hope that was clear for you. I hope that this helped elucidate a little bit of the steps of translation. Any questions, feel free to go back and re-watch the video. Otherwise, enjoy the rest of your day. Thank you. Hi, everybody. Welcome to AP Daily. This is AP Biology, topic 6.4, translation. My name is Lisa Niesman. I'm coming to you from Bay Ridge Preparatory School in Brooklyn, New York. And today we're going to talk about the salient features of the process of translation. The skill we're using today is to focus on predicting causes or effects of a change in a biological system. So let's look at it. We have two big things that we're going to learn today, two questions that we are going to answer. The first is, what are the main features of each step of translation? And second, what are the primary molecules involved in the process of translation? So let's get to it. Translation as a process has many different sequential steps, but before we talk about them, I just want to reorient us, a little review. Remember, translation is the final process in the flow of information from DNA to RNA to protein. The translation step involves converting that RNA information into a protein, just to sort of orient us where we are. So our first step is called initiation. Makes sense. In the initiation step, the rRNA of a ribosome is going to interact with the mRNA at the first start codon. So remember that mRNA nucleotides are grouped together and read in triplets. Each of these triplets is called a codon, and each codon encodes a specific amino acid. So we can tell which amino acid corresponds with which codon by using a codon chart. And when we look at that chart, what we're going to see is that many of those amino acids are encoded for by more than one codon. So let's look at an example of that chart, see what that actually looks like. So this is an example of an mRNA codon chart. They come in different shapes and varieties and sizes, but the key idea is that a chart like this can be used to determine which codon codes for which amino acid. So we have 20 naturally occurring amino acids, which means that some of those are going to be coded for by more than one codon. To look at an example of that, let's take leucine. As you see with the box that just appeared, there are 6 different codons that code for the amino acid leucine. So there's some redundancy there. I also want to point out the start codon, because I mentioned that when we talked about initiation. That codon, AUG, only codes for the amino acid methionine. That's the only spot methionine appears, and that's one way that we know that it's that start codon. I'm going to bring it up now because I have the chart. We also have several codons, the stop codons, that don't actually code for an amino acid. So those are going to be used to terminate this process when we get there. So I said translation involves many steps, and tRNA is really the important molecule that's working here. tRNA's job is to bring the correct amino acid to the correct place as specified by the codon of mRNA. So tRNA has what's called an anticodon, which has to complement the mRNA codon, those base pair, to ensure that the correct amino acid is brought into the ribosome. That's called elongation as that happens. So remember, we saw that met or methionine started the process. From there, the polypeptide is going to get longer, and that's the step process called elongation. So each tRNA, as it brings another amino acid, that amino acid is going to be added to the growing polypeptide chain. The rRNA is going to add the amino acid as the tRNA brings it to the show. This keeps going until we get to the final step, which is termination. So polypeptides could be a variety of different lengths, but we're going to keep going until a stop codon is reached. And once we get that stop signal, translation ends. So there are no more amino acids that are going to be added to that polypeptide. and the polypeptide we just made, that newly synthesized polypeptide, is released. So let's put all of that into practice. Remember, the skill that we're working with today is argumentation. A question is going to ask us to predict the causes or effects of a change in or disruption to one or more components of a biological system based on biological concepts. So let's see what that looks like. Here's a question. I would like you to pause the video, grapple with this question that's asking you to predict. Once you think you have your answer, unpause the video, come back to me, and we'll see how you did. All right, welcome back. The question that we have here is, an mRNA molecule was synthesized incorrectly, and it does not contain a stop codon sequence. Which of the following best predicts the effect on translation if the mRNA molecule is sent to a ribosome? So hopefully you guys read all the way to the end and found answer choice D. The transfer RNA will continue to bring amino acids, and the polypeptide chain will be longer than usual, longer than normal. Without having that stop codon, we're going to continue elongation without termination. So what are our big takeaways from this video? I promised to teach you the salient features of translation, and hopefully I did. The RNA in a ribosome interacts with the start codon of mRNA. That tRNA then brings the correct amino acid to the correct place as specified by that mRNA sequence. Each subsequent amino acid is added to that growing polypeptide chain until we reach that stop codon. And once we reach stop, the polypeptide is released. I also promised to teach you the primary molecules that are involved, and there are a lot of RNAs. The mRNA, which is messenger RNA, tRNA, or transfer RNA, and then R or ribosomal RNA. We're also using those amino acids. So hopefully that cleared things up for you guys. Hopefully the steps of translation make sense. If they don't, feel free to go back and rewatch the video, pause it, and have a great day. Thanks for watching.
Welcome to AP Biology, lesson topic 6.5, regulation of gene expression. My name is Tanea Hibbler, and I teach at Brophy College Preparatory in Phoenix, Arizona. I'm here to let you know that you can do this. Daily video 1. In this lesson, we'll focus on the type of interactions that regulate gene expression, and the skill focus will be on making a scientific claim. What are we going to learn today? We're going to talk about what is gene expression, what types of interactions can regulate gene expression, what is epigenetics, and how is the phenotype of a cell or organism influenced by differential gene expression. Differences in the expression of genes account for phenotypic differences between organisms. Gene expression is a process by which instructions in the DNA are transcribed and translated into a functional protein. The flow of information is always from DNA to RNA to protein. When you go from DNA to RNA, that's transcription. And then when you go from an RNA to a protein, that's called translation. Different types of chemical reactions ultimately are going to regulate gene expression. Regulatory sequences are stretches of DNA that are interacting with proteins. Regulatory sequences are stretches of DNA that can either be used to promote or inhibit protein synthesis. And here we have a regulatory sequence, and we have a gene that's going to be transcribed. You're going to go through transcription and translation to make your protein. Regulatory proteins are used to assist with the promotion or the inhibition of protein synthesis. And the interaction of regulatory sequences with regulatory proteins controls transcription. and it determines what products we're ultimately going to make. Epigenetic changes can affect gene expression as well. Epigenetic changes involve reversible modifications of DNA or histones. And histones are proteins used to wrap DNA around or wrap around DNA. The DNA can either be tightly coiled or it can be untightly coiled. Those slight chemical modifications of DNA and histones cause that tight packing or the loose packing of the DNA. And the packing and the unpacking regulates gene expression. If we can't access the DNA for the gene to be read, then you're not going to transcribe that particular gene and you're not going to make any proteins. Observable cell differentiation results from the expression of genes for tissue-specific proteins. The cells that are within a multicellular organism have the same DNA sequences. So whether we're talking about the muscle cells or blood cells, nerve cells, liver cells, they all have the same DNA sequences within this particular organism. And the tissues are groups of cells that have the same function. The presence of specific proteins within the cells of the tissues give the tissue its particular function. So the muscle cells have one function where the intestinal cells have a different function. The phenotype of a cell or organism is determined by the combination of genes that are expressed, and cell differentiation refers to cells within the same organism having different phenotypes. Induction of transcription factors during development results in sequential gene expression as well. Transcription factors are proteins that promote or inhibit transcription of a gene. And the presence of various transcription factors during development helps determine how a cell differentiates. The process of development from an undifferentiated cell to a differentiated cell is a result of this sequential gene expression. So we start out and there's particular transcription factors in the cell. Those transcription factors can change the cell, and then more transcription factors can further change the cell, and other transcription factors can help the cell become ultimately fully differentiated. Let's go ahead and practice. The lesson skill focus today is argumentation. We're going to make a scientific claim. Go ahead and pause your video right here, read the question, thoroughly study the diagram, and then read the question and determine what's the best answer. Choice A, B, C, or D. Which of the following claims best accounts for the difference in stickleback population phenotypes shown in Figure 1 and Figure 2? And did you pick C? Expression of the PTX1 gene is affected by mutations at other genetic loci. So they have the same gene, but in one of these fish, you're going to see a different phenotype because the regulatory sequence, that regulatory sequence of DNA is mutated, and so that gene cannot be turned on in this individual organism. What should we take away from this lesson? The key takeaways: gene expression is the process by which instructions in the DNA are transcribed and translated into a functional protein. And gene expression is regulated by these interactions that are happening between regulatory proteins, also regulatory sequences, and it's due to the presence of certain transcription factors, or it can be due to modifications of DNA or histones. Epigenetics are reversible modifications of DNA or histones, and they're going to help regulate gene expression. The phenotype of a cell or organism is determined by the combination of genes that are going to be expressed. And we'll talk about it a little bit more in the next video. My name is Tanea Hibbler. Thank you for joining me on this lesson today. And remember, you can do this. Welcome to AP Biology lesson, topic 6.5. My name is Tanea Hibbler, and I teach at Brophy College Preparatory in Phoenix, Arizona. I'm here to let you know that you can do this. Regulation of gene expression. Daily video 2. This lesson will focus on the types of cellular interactions that regulate gene expression in eukaryotes and prokaryotes. What will we be learning today? How does the location of regulatory sequences relate to their function? What's an operon, and why is the lac operon an inducible operon? Both eukaryotes and prokaryotes have genes that are coordinately regulated. Operons are closely linked genes that produce a single messenger RNA molecule during transcription. They're under the control of the same regulatory sequence. It includes the genes to be transcribed, the regulatory sequence, and the operator. An operator is a sequence that either inhibits or promotes transcription by binding with regulatory proteins. In prokaryotes, groups of genes called operons are transcribed in a single messenger RNA. Structural proteins with related functions are typically encoded together in the genome. So they're related to each other, these genes that are going to be transcribed together. They're controlled by a single regulatory sequence. And regulatory genes can control the expression of all the genes at the same time. The lac operon is an example of an inducible operon. It's considered to be inducible because it's usually turned off. When the regulatory protein is bound to the operator, RNA polymerase cannot bind to the regulatory sequence. So here we have a regulatory protein. It's going to be bound to the operator region of this operon. And you see the RNA polymerase that's purple here. It now cannot bind to the regulatory sequence and transcribe this gene. We need something to induce it. This inhibits the transcription of the genes that are part of the lac operon. Like I said, the lac operon is an example of an inducible system. We need something to induce it. Inducers are molecules that can bind to the regulatory protein and cause it to change shape. When the inducer binds to the regulatory protein, the protein changes shape. And in this example, we have allolactose as the inducer. It binds to the regulatory protein, which changes shape, and then it releases from the operator. So this process causes the regulatory protein to release from the operator, and it frees up RNA polymerase to transcribe the operon's genes. So you see here, RNA polymerase is now bound to the regulatory sequence, and these genes that are all located close to each other can be transcribed into a single messenger RNA, and those proteins now can be made. Let's go ahead and look at an example. There are other transcription factors that also can help regulate the lac operon. The amount of glucose in the cell also helps to regulate gene expression. When glucose is low, other transcription factors bind to the regulatory sequence to further promote transcription. And in this example, we have CAMP and CAP, and they're binding to the regulatory sequence, and they're going to promote transcription of the genes. When glucose levels are high, these transcription factors are not present. Let's go ahead and practice. Lesson skill focus is argumentation. We're going to make a scientific claim. Go ahead and pause your video right here. Please read all the diagrams closely. Please read the paragraph and then choose the best answer after reading the question. Which of the following claims best represents the interaction between the repressor protein and the operator? Okay, did you choose C as the best answer? Allolactose, that's a derivative of lactose, it binds to the regulatory protein, which induces transcription. Remember, allolactose prevents that protein from binding to the operator, and so the gene is being freed up to be transcribed. And you can see the other options don't fit in with what we described in the lesson. So what should we take away from this lesson? The key takeaways. Structural proteins with related functions are typically encoded together in the genome. Operons are closely linked genes that produce a single messenger RNA molecule in transcription. And the lac operon is considered an inducible operon because it's usually turned off. Thank you so much for joining me today for this lesson. My name is Tanea Hibbler, and remember, you can do this. Hi, welcome to AP Biology lesson, topic 6.6, gene expression and cell specialization. My name is Tanea Hibbler. I work at Brophy College Preparatory in Phoenix, Arizona, and I'm here to let you know that you can do this. Daily video 1. This lesson will focus on the binding of transcription factors to promoter regions and how this affects gene expression. What will we learn? We're going to learn what promoter sequences are, how the binding of transcription factors to promoter regions affect gene expression and/or the phenotype of the organism. What is the connection between the regulation of gene expression and any phenotypic differences in cells or organisms? How do negative regulatory molecules inhibit gene expression by binding to DNA and blocking transcription? And what role small RNA molecules have in regulating gene expression? So the differences in the expression of genes account for phenotypic differences between organisms. Promoters are DNA sequences upstream of the transcription start site where RNA polymerase and transcription factors bind to initiate transcription. I made the promoter region in this image green, so think of green as go. The interaction between regulatory proteins, regulatory genes, and transcription factors together, those are going to initiate transcription. And so you have the DNA sequence, you have the regulatory gene, and you have transcription factors along with the promoter. And together, that's going to determine how those interactions happen, are going to determine whether that gene is transcribed. The combination of genes expressed determines the phenotype that an organism will have. RNA polymerase may transcribe genes in the presence of transcription factors. But you can have negative regulatory molecules that inhibit gene expression by blocking transcription. So you see on this promoter region, I've added a negative regulatory molecule. So now RNA polymerase can't bind to the promoter. So its RNA polymerase is blocked from binding to the promoter by that negative regulatory molecule. Gene regulation results in differential gene expression, and it influences cell products and function. Cells in the same organism, they're going to have the same DNA, but only certain tissues have activator proteins that can turn on regulatory genes. The phenotype of the cell or organism is determined by the combination of genes that are expressed. Let me go back 1 second here. So in this image here, this is these regulatory proteins are found in the liver. And so this particular gene is going to be transcribed in the liver because these regulatory proteins can activate the regulatory gene, and the interactions that we need with the transcription factors and the promoter will promote RNA polymerase to bind. All of that's necessary to get the products that you need. In addition, certain small molecules have roles in regulating gene expression. Small RNA fragments have roles in gene expression, and they can be encoded by the DNA. They can break down messenger RNA that's in the cytoplasm by recognizing and binding to the messenger RNA. They also can block transcription by binding to the messenger RNA and blocking the ribosome from reading and finishing translation. Let's go ahead and practice. Lesson skill focus is argumentation. Support a claim with evidence from biological principles, concepts, processes, and/or data. Go ahead and pause your video right here. Please look at the image, read the question, look at the answer choices, and choose the answer that you think is best. Which of the following statements best supports the claim that eye tissue cells will transcribe the following gene? Did you choose B? Eye tissue cells contain regulatory proteins that interact with transcription factors to initiate transcription. And so you look at the image here, and these particular proteins are able to interact with this particular regulatory gene. And there's no inhibition going on. And so RNA polymerase can bind to the promoter and transcribe the gene in question. What should we take away? Promoters are sequences upstream of the transcription start site where RNA polymerase and transcription factors bind to initiate transcription. The interaction of promoters and other transcription factors helps determine the phenotypic differences between tissues, within an organism, or between individual organisms. Phenotypic differences in cells and organisms are due to the combination of genes that are expressed. Negative regulatory molecules inhibit gene expression by binding to DNA and blocking transcription. And don't forget that small RNA molecules can regulate gene expression post-transcription by either blocking transcription or by breaking down messenger RNA. Thank you so much for joining me today. My name is Tanea Hibbler, and I want you to remember that you can do this. Welcome to AP Biology lesson, topic 6.7, mutations. My name is Tanea Hibbler. I work at Brophy College Preparatory in Phoenix, Arizona, and I'm here to let you know that you can do this. Daily video 1. In this lesson, we'll focus on various types of mutations and how mutations affect nucleic acids, proteins, and phenotypes. The skill will focus on explaining biological concepts. What will we learn? What a mutation is, what are the various gene mutations, and what effect do mutations have on nucleic acids or proteins and phenotypes. The normal function of the genes and the gene products is collectively comprises the normal function of organisms. When a gene is transcribed and translated, proteins are produced. So the DNA is going to produce messenger RNA, which is transcription, and the messenger RNA produces protein, which is translation. The function and amount of the gene products determine the phenotype of organisms. But changes in the genotype can result in changes in phenotype. Mutations are changes in the genome of an organism. DNA mutations can be positive, negative, or neutral, based on the effect or the lack of effect that they have on the resulting nucleic acid or protein and the phenotype that are conferred by the protein. Whether a mutation is detrimental or beneficial or neutral depends on the environmental context. Disruptions in genes and gene products can result in new phenotypes. Mutations are the primary source of genetic variation that you're going to see within a population. Changes in the genotype can result in changes in phenotype through base substitutions. Gene mutations are changes in the nucleotide sequence of a particular gene. A DNA nucleotide can be accidentally substituted for another nucleotide. So here we have our DNA making messenger RNA, and that messenger RNA is going to produce a sequence of amino acids, and that amino acid is going to produce the protein. Then that's the normal function. If we take this G guanine nucleotide and we substitute it for an A, that's going to change the sequence of the messenger RNA, but it doesn't necessarily lead to a change in the sequence of amino acids or the protein. Let's try doing that somewhere else on the DNA molecule. So if we take the C and we substitute it for an A, that's going to change the messenger RNA sequence. And this time we're going to produce a different amino acid. The last amino acid is going to be different, which can lead to a different protein. These changes can be considered neutral if the change encodes the intended protein. They can be considered positive if the change encodes a new protein that benefits the cell or the organism, or they can be considered negative if the change encodes a new protein that harms the cell or the organism. Alterations in a DNA sequence can lead to change in the type or amount of the protein produced. Changes in genotype can result in changes in phenotype through base insertions and deletions. DNA nucleotides can be deleted from a sequence. DNA nucleotides can be inserted into a sequence. So here we have our DNA and our mature RNA. We make our sequence of amino acids, which produces a protein. Let's say between the A and the G, we insert a base that wasn't there before. So now we have inserted this T base. That's going to change the sequence of the messenger RNA. And in this case, it's going to also change the sequence of the amino acids, producing a stop codon. And so we didn't produce all of the protein that we would have produced previously. We get a different protein. So deletion and insertions shift the order of the gene sequence. This can lead to no protein product or additional protein products. Let's go ahead and look at an example. Sickle cell disease results from a change in base sequences. One base change leads to a change in the messenger RNA and ultimately a difference in the sequence of amino acids. Individuals with this mutated genotype have a different phenotype and their blood may be sickled. The environment, though, is what determines whether this phenotype is going to be detrimental, beneficial, or neutral. So the DNA sequence CTC should produce the messenger RNA GAG, which produces the glucine amino acid. The T is going to be substituted with an A, which is going to change the messenger RNA to a GUG, and that's going to change the sequence. That's going to change the amino acid, and ultimately, that's what produces this different phenotype. Let's practice. Lesson skill focus visual representations. Explain how biological concepts or processes represented visually relate to larger biological principles, concepts, processes, or theories. Go ahead and pause your video right here and be sure to read the picture with the triplet um, numbers and sequence, and go ahead and look at figure 2 as well with the codon chart, and then read the question. A specific protein is determined to confer black spots on ladybugs. The DNA template for the gene encoding for this protein is shown in figure 1. Using the codon chart, explain why changing the triplet number 3 on the template from 3' AAA 5' to 3' AAA 5' will result in black spots. Did you say that the messenger RNA transcribed from the mutated triplet sequence will still code for the same amino acid, phenylalanine, resulting in the same protein? The protein will confer the intended phenotype. So all you have to do is find this particular sequence. You know what uh, what amino acids it's going to code for based on what the messenger RNA would be. And you can see that both of these are both going to code for the same amino acid, so there's no change. What should we take away from this lesson? A mutation is a change in the genome of an organism. Substitutions, deletions, and insertions are gene mutations. And mutations can be positive, negative, or neutral based on the effect or lack of effect that they have on the resulting nucleic acid or protein and the phenotypes that are conferred by the protein. My name is Tanea Hibbler, and I want you to remember that you can do this. Thank you so much for joining me for this lesson. Welcome to today's AP Biology lesson, topic 6.7, mutations. My name is Tanea Hibbler, and I work at Brophy College Preparatory in Phoenix, Arizona, and I'm here to let you know that you can do this. Daily video 2. This lesson will focus on mutations and explain how changes in genotype may result in changes in phenotype. What will we learn? How does a DNA mutation occur? How do errors during mitosis or meiosis result in changes in the phenotype of a cell or organism? And what determines whether a mutation is detrimental, beneficial, or neutral? The processing of genetic information is imperfect and is a source of genetic variation. Disruptions in genes and gene products cause new phenotypes. Random mutations can be caused by each of the following: errors in DNA or replication, errors in DNA repair, radiation, reactive chemicals, and mutations are the primary source of genetic variation. Changes in chromosome number often result in new phenotypes. Errors in mitosis or meiosis can result in changes in phenotype. Chromosomes may not separate properly during cell division. Triploidy is having 3 copies of a particular chromosome. It can result in sterility in some animals. Polyploidy is having multiple sets of homologous chromosomes, and it can result in increased vigor in plants. Changes in chromosome number often result in human disorders with developmental limitations. Improper separation of the chromosomes can lead to gametes with missing or extra chromosomes. Trisomy 21, also notated as 47XX plus 21 or 47XY plus 21, results when you have an extra chromosome at chromosome number 21. And Turner syndrome, 45XO, results when you have a missing X chromosome. So we can have let's say you have your cells, and your sex chromosomes are supposed to separate when you're going to make your gametes. Well, if they don't separate properly, you end up with gametes that either have too many sex chromosomes or they're missing their sex chromosomes. If those are the gametes that are going to be used during fertilization, then you can end up with a disorder like this, which is Turner syndrome. And looking at this karyotype, we can see here that this individual is missing an X chromosome. Whether a mutation is detrimental, beneficial, or neutral depends on the environmental context. The environment is not static. Changes in the environment can impact survival of organisms. A particular phenotype can be beneficial under certain environmental conditions and detrimental under other environmental conditions. So we see here when the habitat looks one way, you can have a beneficial trait. With that same organism, under other conditions, that trait can be detrimental for them. Let's look at an example. Polyploidy in plants and sterility. Polyploidy can also result in sterility. The Cavendish bananas are triploids. That's a tetraploid that's mated with a diploid. And I have this image over here so you can see the 2N gamete comes together with the N gamete, resulting in a triploid offspring. These polyploids are infertile. They can't reproduce or make seeds. Another example is polyploidy in plants. They can have increased vigor. These strawberries, they look fairly normal. These strawberries look gigantic. So in plants, this often results in increased vigor. These strawberries are phenotypically larger than normal. What should we take away? Mutations in DNA can be caused from errors in replication, repair, or radiation or chemical exposure. Errors in mitosis can change the number of chromosomes in an organism, which can result in increased vigor in plants, sterility, or disorders in humans. And the environment determines if a mutation is detrimental, beneficial, or neutral. My name is Tanea Hibbler. Remember, you can do this. And thank you for joining us today with this lesson. Welcome to today's AP Biology lesson, topic 6.7, mutations. My name is Tanea Hibbler, and I teach at Brophy College Preparatory here in Phoenix, Arizona. I'm here to let you know that you can do this. Daily Video 3. This lesson will focus on explaining how alterations in DNA contribute to variation that can be subject to natural selection. The skill will focus on making observations. What will we learn? How are changes in genotype subject to natural selection? What are horizontal acquisitions of genetic information? And what reproduction processes increase genetic variation? Changes in genotype may affect phenotypes that are subject to natural selection. Natural selection is a process in which organisms better adapted to a particular environment have a higher chance of survival and reproduction, thereby passing on those adaptations to the next generation. Genetic changes that enhance survival and reproduction can be selected for by environmental conditions. The horizontal acquisition of genetic information increases variation. Horizontal transfer of genetic information refers to the exchange of genetic information between different genomes or between unrelated organisms. This primarily occurs in prokaryotes. It contributes to genetic variation in prokaryotic populations. This includes transformation, transduction, conjugation, and transposition, of which we'll look at some examples. The horizontal acquisition of genetic information in prokaryotes increases variation. Transformation refers to the uptake of naked DNA. Naked DNA is DNA not protected by proteins or other molecules. So DNA comes from an external environmental source. So this prokaryotic cell was able to take up this naked DNA. This occurs primarily within prokaryotic organisms. The horizontal acquisition of genetic information increases variation. Transduction is a transmission of foreign DNA into a cell when a viral genome integrates with the host genome. So that virus is going to bring some genetic information to that bacterial cell. And now that information is going to be incorporated within that bacterial cell. The horizontal acquisition of genetic information increases variation. Conjugation is a cell-to-cell transfer of DNA. This allows for small segments of DNA to move from one cell to another. So one bacterial cell is transferring information to another bacterial cell. An external cell extension connects the cells and allows for this transfer. And this also primarily occurs in prokaryotic cells, as we see here in this example. The horizontal acquisition of genetic information increases variation, and transposition is the movement of DNA segments within and between DNA molecules. This can happen in many different ways. You can see that pieces can be moved from one chromosome to another, or deleted or duplicated, or inverted. Related viruses can combine genetic information as well. More than one virus can infect the same cell. Viral genomes can combine once they're integrated into the host genome. So here we have virus 1 and we have virus 2. They have both infected the same cell, and their information is going to be integrated into the genome of the host cell. And so when they go to produce more viruses, you can actually produce a virus that's new. It's a combination of virus 1 and virus 2. And we can see here in virus 3 that we have genetic information from both of those viruses. So new viruses can have the new viral combinations. Reproduction processes can increase genetic variation as well. Certain reproductive processes are evolutionary conserved, and they're shared by various organisms. Sexual reproduction and the combination of random gametes is one of the ways that we increase genetic variation. You never know which sperm and which egg are going to combine and which genes they have, and so we get all types of new combinations as a result of sexual reproduction. We also have independent assortment of homologous pairs. You don't know which chromosome is going to line up on which side of the cell during meiosis. And so we can get all types of interesting combinations because we don't know how they're going to assort, and they're assorting independently from each other. And crossing over during meiosis also is going to increase genetic variation when you have segments of the chromosome that are swapped. Then on homologous pairs during crossing over, you actually get new combinations of genes. So maybe the big A, little b combination is a new combination that wasn't seen before. Let's go ahead and practice. Lesson skill focus, questions and methods. Make observations or collect data from representations of laboratory setups or results. Go ahead and pause the video here. Please read the question thoroughly. Please look at the image and then look at your answer options. Which of the following would most likely be observed in plate 2 after 24 hours? Did you pick choice D? The answer says there would not be a clear ring around either disc A or disc B. And I think it's really important that you go back to the paragraph and you notice it says a single bacterial colony is observed with physical characteristics like those of the pure culture. So it should be behaving like all the rest of the cells in the pure culture. And so that's why you're not going to see a clear ring around either disc A or disc P. What should we take away? Genetic changes that enhance survival and reproduction can be selected for based on specific environmental conditions. Horizontal acquisitions of genetic information include the transfer of DNA segments between cells, viruses, and cells, or movement of DNA sequences with and between DNA segments. And genetic variation is increased through sexual reproduction, independent assortment of chromosomes, and crossing over. My name's Tanae Hibbler. Remember, you can do this. Thank you so much for joining me for this lesson. Welcome to today's AP Biology lesson, topic 6.8, Biotechnology. My name is Tanae Hibbler. I teach at Brophy College Preparatory here in Phoenix, Arizona, and I'm here to let you know that you can do this. Daily Video 1. This lesson will focus on techniques used to analyze or manipulate genomes. The skill focus is to explain relationships between experimental results and biological concepts. What are we going to learn in this video? What are some common techniques used to analyze or manipulate DNA and RNA? And how can genetic engineering techniques be used to manipulate DNA and RNA? Genetic engineering techniques can be used to analyze and manipulate both DNA and RNA. Common techniques used to analyze DNA and RNA include the following: electrophoresis, polymerase chain reaction, PCR, DNA sequencing, and in gel electrophoresis, it separates molecules according to the size and charge. DNA molecules are negatively charged molecules. DNA samples containing fragments can be loaded into wells filled with a gel. You can have a current applied to the gel, creating positive and negative ends. And the DNA is going to move towards the positive end of the gel. It's quite fascinating, actually. This separates DNA fragments by size, and smaller particles will move faster and further through the gel. And you end up with bands that indicate molecules that migrate to that location in the gel. And so you can compare different samples in this way. And these band samples can be used also to identify people. During polymerase chain reaction, PCR, DNA fragments are amplified. So PCR techniques allow scientists to create large samples of DNA to analyze when just a small sample is initially available. Technique involves several steps. First, you have to denature the DNA. So that's what we're doing here. We're denaturing it. We're separating those bonds between the nucleotides. And then we're going to add primers. And that's like a sequence, basically. It's going to allow the copying strands that we want to put on there so they know where to start. And then we're going to replicate that DNA. And so now we have twice as much DNA as we started. And this process can be repeated until you have an ample amount of DNA to do whatever analysis that you want to do on it. Bacterial transformation introduces DNA into bacterial cells. So here we have a bacterial cell and we have some foreign DNA. Transformation is a process by which that bacterial cell takes up foreign DNA from external sources. Bacterial cells only uptake foreign DNA at specific times. The DNA can be incorporated into the bacterial chromosome, or it can exist separately into the cytosol. So that DNA is taken up, and here I'm showing an example of how that DNA has been incorporated into the bacterial chromosome. And then here I'm showing how that DNA has been incorporated into the cytosol as a plasmid. So scientists introduce foreign DNA during the time that bacterial cells are primed for uptake. This can be used to make medicines, you can modify food this way, or you can amplify DNA. DNA sequencing determines the order of nucleotides in a DNA molecule. And with the technology that we have available now to us, we have many ways that we can do this. These techniques are establish the order of nucleotides in a strand of DNA molecules. There's numerous techniques employed to sequence DNA, but this includes nucleotides being labeled with fluorescent dye to read and build copies of DNA. DNA runs through a capillary gel, and a detector will read the sequence. So all the different DNA pieces that are different sizes with the labeled nucleotides are going to be run through, and the smaller fragments are going to come first, and they're going to separate by size. And so we end up determining what the sequence of DNA is. And you can see in the picture there's a laser and detector that can identify what those nucleotide bases are. DNA sequencing can be very important in evolutionary biology, medicine, and forensics. Let's go ahead and practice. Lesson skill focus, argumentation. Explain the relationship between experimental results and larger biological concepts, processes, or theories. Go ahead and pause your video here. Please look at the diagram, read the figure, read the paragraph, and look at your answer choices. Which of the following best explains why there is no growth on plate 2? Did you pick answer A? The initial E. coli culture was not ampicillin resistant. So because it's not ampicillin resistant, when you grow it in the presence of ampicillin, it's going to kill all of those bacterial cells. So what should we take away? Electrophoresis, PCR, bacterial transformation, and DNA sequencing are used to analyze or manipulate DNA and RNA. And genetic engineering techniques can be used to amplify, modify, and compare samples of DNA and RNA. Thank you so much for joining. My name is Tanea Hibbler, and I want to remind you that you can do this.